Hi, this video is on Orthomyxovirus, which comes under the superfamily Myxovirus, right? And if I say Orthomyxovirus, you won't get it correctly. But if I say influenza virus, that's a very common member of Orthomyxovirus family. Swine flu, bird flu, and so many SARS and all those kind of respiratory associated virus comes under this Orthomyxovirus superfamily. So before jumping into the actual topic, let's learn something about their family characters and their basic features, and then we'll learn about influenza virus and their features. Now, talking about orthomyxovirus comes under the superfamily Myxovirus, right? That's the name of the superfamily, and where myxo or myxa means the mucus. So myxovirus are a group of virus that binds to the mucin receptors on the surface of RBCs, and they also show some kind of connection towards the mucous membrane. So these are classes of virus which shows a kind of high rate of sensitivity or high rate of tropism towards mucin and mucosal membrane. Now, myxovirus, all of them, they have the capacity to bind even with the mucin receptors of RBC. Then that sounds really dangerous because they can combine with RBC. So definitely they can cause agglutination, hemagglutination, or clumping of RBCs that can block important blood vessels. So this myxovirus comprises of two different types of subfamilies, orthomyxovirus and paramyxovirus. So myxoviridae or Myxaviridae is a superfamily in which we have two subfamilies, Orthomyxoviridae and Paramyxoviridae. Among this list, we will be mainly focusing on Orthomyxoviridae in this presentation. So let's compare the characteristic features of Orthomyxovirus from Paramyxovirus. So the size, Paramyxovirus are quite bigger in size. The dimensions for Orthomyxovirus is ranging between 80 to 120 nanometer. In paramyxovirus, it is 100 to 300 nanometer. Orthomyxovirus is more spherical in nature. Sometimes it can also appear in the filamentous format in case of high rate of infections. Usually paramyxovirus shows pleomorphism. They won't stick to one particular shape. They always follow a complex symmetry in terms of their structure. And one common feature for both of them is they consist of RNA, single-stranded RNA as a genetic material. But even though they have the same genetic material, same kind of genetic material, in case of orthomyxovirus, it is not a continuous RNA, it's more segmented. They got segmented RNA, which almost comprises of 8 pieces. They got segmented RNA or pieces of RNA is present in the nucleocapsid. I'm talking about orthomyxovirus. Whereas in paramyxovirus, it's more linear, single or a single strand of RNA virus. And the multiplication for orthomyxovirus or viral replication for orthomyxovirus happens within the nucleus, whereas paramyxovirus replication happens at the cytoplasm of the host cell body. And genetic reassortment is really common in orthomyxovirus. Genetic reassortment means, like I said, influenza virus. Every time when they appear as a pandemic or epidemic, they'll come in a new combination. Hope you remember H1N1, H1N5, H2N1. So these are different combinations, genetic combinations. So orthomyxovirus can do a genetic reassortment, whereas paramyxovirus will not be able to do that. So orthomyxovirus are really dangerous in comparison with paramyxovirus. And since orthomyxovirus can do this genetic reassortment, we can't provide a specific vaccination for all the varieties. We can't generalize into the prophylactic measures. Right. So the next point, antigenic stability. Orthomyxovirus are absolutely variable in terms of their antigenic stability. They won't show any kind of antigenic stability. They keep on changing. Whereas paramyxovirus is somewhat stable, so somehow we can manage paramyxoviral infections. Example for orthomyxovirus is influenza virus, the one which we will be learning in this presentation. An example for paramyxovirus includes the virus which can cause parainfluenza, mumps, measles, respiratory syncytial virus. All those are coming under paramyxoviral classification. So let's talk more about Orthomyxoviridae or Orthomyxovirus. The commonest member and the most dangerous pathogenic member of Orthomyxovirus is, of course, the influenza virus. They can cause serious respiratory tract infections in human body. That's why it's called viral influenza or flu kind of virus. It consists of three types of genera. I mean, influenza virus are of mainly three different types. We have named them as group A, group B, and C. So this is the major genus of of the family Orthomyxoviridae. Influenza virus are of three different types: influenza virus type A, influenza virus type B, influenza virus type C. This is the genus variation, but among this genus variation, they show different antigenic sub variations. And in this slide we can make out the examples for few other Paramyxoviridae, which includes Newcastle disease virus, mumps virus, parainfluenza virus, virus which cause measles, and virus which can cause respiratory syncytial formation. Syncytium means joining of, clumping of adjacent cells to form a single large cell, multinucleated large cell. That's the specialty of Paramyxovirus. Now, let's talk about the influenza virus in detail. Let's begin with the morphology of this virus. This virus is more spherical in nature. Sometimes they can also appear in the filamentous format. Right, and the size ranges between 80 to 120 nanometer, and they have a helical symmetry. Usually they contain segments of RNA. If you start learning from the core part, they got segments of RNA, and this RNA is attached with the nuclear protein, ribonuclear protein, with helical symmetry, which is single stranded but segmented pieces of RNA. And since this pieces of RNAs are there, they can recombine in any format. They can recombine with another genus or another variety of influenza virus. That's how they makes this antigenic variation. They got RNA polymerase enzyme because that is required for the translation process and also for the conversion of viral RNA to viral DNA. They are enveloped virus. Of course, they are enveloped virus that they got enveloped part from the phospholipid bilayer membrane from the host cell itself, so that they can easily escape from host immune system. They also got peplomiyas. Peplomiyas means surface receptors, which can enable them to show cell tropism and also show hemagglutination and dissociation. I mean, they show two kind of peplomiyas. One is hemagglutinin spikes, which shows hemagglutination reaction, and neuraminidase, which has some enzymatic activity that we'll see in the next slide. And replication is usually one Magnus phenomenon. One Magnus phenomenon means usually abortive replication. They replicate in high numbers, and that always ends with some kind of issues, and they produces abnormal numbers of immature viral particles. So let's see the morphology of this virus. It is more spherical. Sometimes they can also appear in the format of helical symmetry, right? And they consist of segments of RNA. These are the RNA molecules, but in the segments format, 8 pieces of 8 segments of RNAs are present, which is covered by the capsid protein, right, which is composed of capsid, which is composed of capsomeres, and this capsid is again protected by the phospholipid bilayer membrane. That's how the virus becomes enveloped, and that is something which the virus gets from the host cell itself, right? On the surface of this envelope, the virus has got two different types of peplomiyas or glycoprotein spikes, and one is N spike, or that is called neuraminidase spikes, and H spikes, which stands for hemagglutinin spikes, and we'll see the actual functions of these things in detail in the following slides. Right. This is another image, a simplified image of influenza virus. They got matrix protein also, same like HIV virus, which is covering the capsid, and they got envelope, which is composed of hemagglutinin and neuraminidase, spikes. So this is how the virus multiplies within the host cell. They follow the normal. This is almost same, just like any other virus. They follow adsorption, penetration, uncoating, biosynthesis, assembly, and release. Everything is same. And since they're RNA virus, they can take control of they can make use of RNA polymerase enzyme to integrate their viral DNA after conversion into the host genome, and also to help in translation process to produce new viral particles. And after assembly, they are usually naked particles. They are not enveloped, but at the time of budding, they'll come out by taking some amount of host cell membrane, and they'll become enveloped virus. Now, talking about the antigenic structure, they have a pair of internal antigens which are usually known as ribonucleoproteins, and these ribonucleoproteins are of three different types A, B, C, and based on that they got three types of influenza virus: influenza type A, influenza type B, and influenza type C virus. But more than this internal antigen, the most significant ones are the surface antigen, which is present on the surface of the, or projecting from the surface of this virus. They got M protein. This is something which is covering the capsid portion, which forms the matrix, and their main function is to provide protection, extra protection from host defense mechanisms. And apart from this M protein, they also got V antigen, which has two virus encoded proteins, hemagglutinins and neuraminases. And this is the important function of hemagglutination and neuraminidase spikes. Hemagglutination spikes will definitely bind with mucoprotein receptors on the cell membranes of RBC. So many number of RBCs can clump together. So that's how that's why the influenza become really dangerous. It can go serious clumping around the RBCs, and so too many RBCs will come and clump around these viral particles, and even viral infected RBCs will become viral infected RBCs will become more sticky to bind with the neighboring RBCs, and that's how they create a super agglutinin clumps, blocking the blood vessels. And neuraminidase is another important thing, which is for the elution purpose, receptors destroying the enzyme. Receptor destroying enzyme, which produced by some bacteria like Vibrio cholerae. So that's actually a survival strategy for the virus. So imagine influenza virus entered a healthy individual, and it started clumping around the RBCs, and there is imagine there is a mass of agglutination happened there. Now, if the person start taking medication, antiviral drugs, or maybe interferon therapy or any kind of vaccination, now this virus can easily sense it, and they knew that yes, if I stick with RBCs now, I will be in trouble. So what they will do, they will activate neuraminases, and neuraminases will break the connection between hemagglutinin spikes and RBCs, so that this influenza virus is now free to be transported along with blood circulation to a new site where they can hide out, for a longer period of time. That's actually how they have contradictory activities. Hemagglutination help the virus to attach with the RBC, and neuraminidase will separate this attachment. So that's how it happens, right? So hemagglutination, we have made use of this hemagglutination property for lab diagnosis purpose, right? We have used this technique for lab diagnosis. Hemagglutination provides convenient method for detection of and titration of virus in egg and other culture fluids. So we can detect the presence or absence of influenza virus just by mixing it with RBCs, and we can also have an idea about the titer values. Titer values means amount of virus which may be present in the patient sample. So hemagglutination titer is the highest. We will go for serial dilution. We'll go for serial dilution of patient sample, on which we'll add a specific amount of RBCs. Hemagglutination titer is the highest dilution of virus suspension that produces hemagglutination of a fixed quantity of cells. So it's quite simple, just like a viral test, where we will keep a series of test tubes. And in these test tubes, we will add virus suspension or the patient sample in increasing dilutions. So the first tube will have high dilution, not high dilution. The first tube will have high quantity of virus, and towards the last test tube, the amount of virus will decrease, or towards the final dilution, the viral load should increase. And in all this test tube, we will add equal amount of RBC suspension. And if this patient sample is having influenza virus, definitely they will show hemagglutination that can confirm this viral infection. And imagine the agglutination happens even till the last test tube. That means there is that many number of viruses present till the last test tube that is causing hemagglutination. That shows a kind of predictory method. So we can confirm the infective condition on this individual. So highest dilution, if it shows a positive result till the highest dilution, that means the person is severely infected, right? That's how we made use of this technique, hemagglutination. Now, hemagglutination inhibition is the highest dilution of serum that inhibits hemagglutination. So we also use hemagglutination inhibition analysis for the detection and quantitation of antibodies to virus. So if there is hemagglutination inhibition in the highest dilution, that means the serum sample, the collected serum sample is having high amount of antibody. That means the person is super immune to or super active against this viral infection. But the major disadvantage of hemagglutination inhibition is presence of non-specific inhibitors. We don't know what is actually inhibiting this hemagglutination. It can be because of the actual antibodies, or maybe something else like trypsin, potassium peroxide, periodate and coalin, and even carbon dioxide can inhibit hemagglutination. So we won't be able to confirm the presence of antibodies, but we can assume, yes, if it is after having any vaccination, we can think that yes, vaccination is working. So this is hemagglutination inhibition. It's again the same process. We'll do serial dilution of patient serum sample, and we'll add equal amount of RBCs. So along with hemagglutination, they can also do heme adsorption. Heme adsorption is the non-specific response where large number of RBCs will clump together around a cells. This actually happens to those. This actually happens around those RBCs which is actually infected by the influenza virus. So the plasma membrane of tissue culture cells in which the virus is multiplying, containing hemagglutinin. So around the host cells, new cells will come and join. So agglutination or hemagglutination is actually happening between the virus and RBC. Heme adsorption actually happens between virus infected cells, host cells, with other healthy cells. That's the difference, right? So red cells are adsorbed on the surface of such cells, and advantage is we can use hemagglutination process to confirm the growth of virus in tissue culture media. And this is how they may look like under the microscopy, heme adsorption. Now, the major problem with influenza virus or Orthomyxovirus is that they show various type of antigenic differences or antigenic variation, and we use two pattern of antigenic variation is shown by this virus. They are known as antigenic drift, and the other one is antigenic shift. So these are varieties or categories of antigenic variation shown by this class of virus: antigenic drift and antigenic shift. So if you talk about antigenic drift, it's a gradual sequential changes in antigenic structure which happens at frequent intervals. That's quite predictable. We know that yes, this can happen within 10 years of time or 5 years time. That's a gradual change. If you constantly observe this virus, we'll have an idea, okay, what will be the next combination. Whereas antigenic shift is abrupt, drastic and discontinuous. That is absolutely new. We will not have any clue. We'll never have any clue that what kind of combination is going to happen next. Right, and antigenic drift is different from the previous, but it is quite related. It will be different, but it will have some connection with the previous earlier viral or parental viral particles, whereas antigenic shift is completely different from the parent one. So imagine I produced vaccine for H1N1 virus, but in the next epidemic it will come as H1N2. So the vaccine I produced for H1N1 will be useless for H1N2. That's a new variety, so it's completely different. So antigenic drift usually happens due to mutations and selection because of periodic epidemics, and it's a natural selection process. But antigenic shift is happening due to genetic reassortment because they hold fragments of RNA particles, so they can randomize, so they can change their arrangement in whichever way they want, and they can go antigenic shift. So antigenic shift is obviously more dangerous than antigenic drift. I think we have an image. Yes. So this is antigenic shift. Antigenic shift is usually, so this is one virus, one influenza virus, which shows combinations of hemagglutinin and neuraminidase spikes. This is another virus, and during antigenic shift, these two things will join together to form new combinations. So here we have one combination, here we have another combination. These two things will be completely different from their host body. You can see this is the genetic makeup of virus A, and this is genetic makeup of virus B, but see the genetic arrangement of these products is completely different, and even the arrangement is, they can be arranged in any way, in whichever way, so it's completely different from the parent cell. Right. Talking about antigenic drift, this is happening within the single viral particle. So this is happening at gradual intervals. It's a slow process. So we have some kind of idea. We can assume that what kind of combination it can produce. So antigenic drift is quite predictable, whereas antigenic shift is unpredictable. This causes drastic, serious complications in the human population. Right. So these are some of the influenza virus subtypes that occurred over the years. We got H1N1 virus, H2N2 virus, H3N2 virus. H stands for hemagglutinin and N stands for neuraminidase, and they show slight variations in terms of their virulence factors or viral markers, and they come up in new combinations every year. So we can't predict what will be the next combination. So it came up like causing a swine flu, like pigs were causing the transmission of infection. Even birds can cause the transmission of infection. Then we use the term bird flu. So the virus is same, the basic structure is same, but it comes in a different antigenic combinations. Now, talking about the pathogenesis of this virus, like I said, this mainly causes respiratory tract infections. So the main route of entry will be person-to-person contact. Patients can act as the correct source of infection. Sometimes it can also come to human body through animals. So protective mechanism is whenever the virus enters, there will be cough reflex, initiation of cough reflexes, and mucosal Ig antibodies will try to destroy them, but it's unsuccessful because the virus has got Ig antibodies will not identify them as foreign particles because the envelope is same from human host cell itself. And mucous secretion can also do their job, but it's unsuccessful in immune suppressed or immune compromised individual. See the incubation period within 4 to 5 days, the person will start showing specific symptoms. So there will be initial viral destruction if the person is strong, but neuraminidase lowers the viscosity to mucous film and promotes the spread of virus to deep layers of or deep inner layers of respiratory tract. And once it reaches the lower layers of the respiratory tract, there will be desquamation of superficial mucosa and the respiratory tract, and this can bring secondary bacterial infections, which is usually caused by staphylococcal, streptococci, even Haemophilus influenza bacteria. So this is one major reason which this is something which will complicate the entire scenario, causing serious issues, life-threatening complications in the even in the healthy individuals. So these are the common symptoms of swine flu. Everything starts with systemic complication like fever. There will be lethargy, lack of appetite, runny nose, sore throat. These are our response, like our immune system will try to eliminate this virus from human body, and there will be respiratory tract complications like coughing. There will be intestinal complications like diarrhea, and there will be gastric issues with the nausea and vomiting. These are some of the symptoms associated with influenza virus. And these are the 3 clinical features which usually happens in immune compromised or immune deficient individuals. There will be uncomplicated influenza, there will be pneumonia, and there will be Reyes syndrome. We'll see all these things in detail. So uncomplicated influenza, as the name suggests, it's not a serious condition. Everything starts with a fever, headache, generalized myalgia, and abdominal pain. These are the earliest clinical presentation. So uncomplicated disease will resolve within 7 days. That's a common case. But if it persists, it will lead to the next phase of this infection, that is Reyes syndrome. Uncomplicated influenza will develop into pneumonia condition. We know what is pneumonia means. There will be an extreme mucus accumulation in the lungs, causing serious respiratory distress syndromes and all, but it can also lead to Reyes syndrome. It's a fatty degeneration of liver with acute encephalopathy occurring in the children and adolescents cases, and it is the actual reason for Reyes syndrome is still unknown. It may be because of some hyperactive allergic response which is created by the virus itself, and the mortality rate is 10 to 40%. So if this influenza condition can develop to Reyes syndrome, that will be really dangerous to the individuals. So coming to the lab diagnosis, we have lab diagnosis of influenza virus, and this is based on 3 options. One is demonstration of virus itself. Of course, we need to go check the samples under the microscopy. We can isolate the virus in lab conditions, and of course we can do some serological examination. Serological examination is quite easy because demonstration of virus under lab conditions definitely demand the requirement of high intensity electron microscopes in the lab. So demonstration of virus, we can demonstrate the presence of virus by direct immunofluorescence or polymerase chain reaction. For direct immunofluorescence, definitely we need an immunofluorescent microscope, and we can also do the polymerase chain reaction to track down the viral RNA particles. And talking about the advantages, it's quite rapid and it's detected in nasopharyngeal cells. So nasopharyngeal swabs can be used as a sample, right? And isolation of virus is possible if the sample has been taken within 2 to 3 days of infection, because it's usually present in the upper layers. Throat garglings with buffered saline is the best sample. We can use embryonic X for isolation of virus, and we can even use cell cultures to isolate virus. So we can grow them in amniotic cavity, 11 to 13 years old egg embryos, which is incubated at 37 degrees Celsius for 3 days, and amniotic fluid is tested for hemagglutination. In guinea pig, type A will be tested in guinea pigs RBCs, and type B can be performed in guinea pigs and fowls RBC, whereas type C is only for the fowls RBC. Right. We can do primary and continuous cell cultures for the identification of influenza virus, and confirmation is again done by hemadsorption with the human O group RBCs. But the most useful and the most easiest method is, of course, doing the serological examination, where we can start doing with complement fixation test, which is type specific, or we can do hemagglutination inhibition test, which is subtype specific, and hemagglutination inhibition test provides a clear-cut idea about the presence of protective antibodies in patients' serum sample. So as the dilution increases, that shows if it is hemagglutination test, if the dilution increases, that means the person is severely affected. In case of hemagglutination inhibition test, as the dilution increases, that means the person is having protective antibodies. And I was talking about this one, genetic reassortments, antigenic drift. In 1918 it was Spanish influenza with H1N1 combination. In 1957 it was H2N2 influenza, which is called Asian influenza. In 1968 it was H3N2. Recently we had H1N5 type variety. So next pandemic, it's unpredictable because of this antigenic shift, which is usually happening in influenza virus. Talking about the treatment options, we have different antiviral drugs available, which will primarily suppress the viral multiplication, and it can support immunoglobulin A, which is present on the mucosal layers, which can also protect the M2 proteins, which is involved in the proper binding of the mucin layers. So these are the commonly used antiviral drugs. Now, talking about the prophylaxis, major indications, especially in cold countries and pandemic threat areas, definitely we have to think about prophylactic measures. Of course, we are not so successful in providing a specific vaccination against all those types of influenza, because they keep on undergoing these kind of changes. So we have successfully made different types of vaccines which can provide a generalized protection against influenza virus, something which has been produced by inoculating them in allantoic membrane of embryonic eggs, and we have used a subunit vaccine to reduce toxicity, and we have even used recombinant vaccines and live vaccines also. But their activity is limited because in every pandemic, it will take a longer time to produce specific vaccination. And we can better prevent this human and human-to-human transmission by keeping the mask, something which we currently practice in our day-to-day life to prevent coronavirus infection. Frequent washing of hands, and avoiding the touching of surfaces, and avoiding close contact. All these are common general practices by which we can prevent the transmission of infection. Right. That's all about influenza virus.